Hello, welcome to this session on how nerves make signals. This is part of the aspects of human physiology unit, but it links with so many other things. And today what we'll be explaining is how a nerve makes a signal. How do we send a message to our muscles to make it contract? How do we send a message to our heart, for example, to tell it to speed up or slow down? To me, it's a miracle. It's absolutely fascinating to learn, and I hope you find it really useful. So in terms of what we're going to do today, we will be learning about how the nerve generates a signal. So where does it come from? How does it start? And we've learned already that the travel on a neuron goes from the cell body to the terminals, or the soma to the terminals. It only goes one way, so we'll be looking at how that starts in the soma, or the cell body, and then how it propagates along the length of a cell. So propagates is just a posh word for travels along, or passes along. So how does it go from the cell body to the terminals? And hopefully, as a result of that, we should be able to link the knowledge to pharmacology, so maybe to explain how anesthetics work, how ibuprofen works, or paracetamol works, and other possible neurotoxins that we might have come across. So we've come across this cell before. It's a neuron, and as I said in the previous slide, the signal travels from the soma to the terminals, or the head, the cell body of the terminals. So this signal generates around this part, is then channeled into this area and sent down this orange tube to the terminals, where it will then meet probably either an effector, such as a gland or a muscle, or another neuron, which then another neuron will fire off. So this is the end, the terminal ends, the last place where the signal goes to. And you've kind of learned in terms of a muscle, or you should know by now, our skeleton and muscles unit, that if it's a motor nerve attached to a muscle, then this place here would be the neuromuscular junction and the synapse. Pretty much the synapse, the gap between the nerve and the muscle. Then we have these rods, these myofibrils running with said lines and all this kind of stuff, which would contract when a nervous impulse is sent. Okay, hopefully that made a bit of sense. So, when learning about how a nervous impulse is propagated and created, there are 4 things that you need to know about, and they're on the screen right there. Resting potential, graded potential, action potential, and saltatory conduction. They're the 4 things. Shortened it down to our gas. You need to learn about our gas, our brother gas. And once you know about those 4 things, you should be able to explain what's going on. So before we even get into it, I just want to sort of summarize what they are. So you can come back to this slide and it will tell you a bit about what it is if you need to go back to the start, kind of thing. Resting potential refers to the charge at rest across a membrane. So if we had a membrane here on the right hand side, now obviously this is separated between the inside of the cell and the outside of the cell. So the resting potential is a measurement of charge across a membrane at any one point. Okay, so it might be positive on this side and less positive on that side, and it's just a measure of how positive or how negative it is. And I'll explain that in more detail, but that's basically the principle. So I guess if we had a classroom here, this is the inside of the classroom, this is the outside of the classroom. I'm just trying to think of an analogy that I might want to use. We've got lots of students outside this classroom wanting to get in to the classroom. All right, that's the potential. So if the door's closed, That's what we mean by potential. It has the potential to move. It has the potential to move from this side to this side, and that's where we're talking about potential. It's sort of a term that's banded towards electricity quite a lot. It's the potential of something to move. So if we open this door to the classroom, we have the potential of a lot of students filling this room. Whereas if we had a classroom such like this, and we had 2 students outside waiting to get in, as soon as you open the door, they will flood in, but not with any sort of, not with the same power as this classroom here that's got a lot of students wanting to get in. So the potential is lower on this side than it is on this side, and that's what potential means in terms of sort of electricity, because that's kind of what we're talking about. It's not exactly the same as electricity that comes through your plugs and your electrical devices, but it is similar. Potential is the potential movement. It's the potential movement. So if we've got a lot of something on one side and we've shut the door, then this side has a lot of potential. You know, if we open the door, it's going to flood in. Whereas if we have less things on one side, then it has less potential. Okay. Now that's at rest. So a neuron, a resting potential, is the charge across the membrane at rest. So these 2 potentials that are next, the graded and action, are the 2 sort of, they're the 2 processes that are involved in the actual signaling of the message. The graded potential is what kicks us off. Okay, it's what kicks us off, and then it switches to an action potential, and then it goes one to the other. So actually, when we look at the neuron, if I draw the kind of neuron here, very rough, but hopefully you get the idea. Here's some dendrites off it, and we have the myelin sheath. Okay, it will start with a graded potential, and then it will turn to an action potential, and then wherever you get this myelin sheath, it will turn back to a graded action, because this is a node of Ranvier. This is not myelinated, it's not got myelin around it, and then wherever there's a myelin sheath, it'll go back to a graded potential, and then it'll go back to an action potential. There is a real key reason for this, which is hopefully what I'm going to explain, but I just want to give you a basic rundown before we get into it, so that you can see the big picture before we narrow it down into the smaller picture. So when a nervous impulse fires off from the soma to the terminals, it starts with a graded, then turns to an action potential here, and then wherever there's a myelin sheath, it will turn to a graded potential, and then when there's not a myelin sheath, i.e. these nodes of Ranvier, in between the myelin sheaths, turns back to an action potential, then graded, then action, and then it'll hopefully pass on to wherever it needs to go. That's the 2 potentials that are doing the work in a nervous impulse. Finally, saltatory conduction. Saltatory conduction is just a term that describes this, i.e. going from a graded to an action, to a graded to an action, to a graded to an action along the axon. So this is just a term that describes these 2 alternating from one to the other. So actually it's not a process on its own, it's just a combination of the processes. So really these 3 things are going to be the most important things to understand. What is charge? What is how does a neuron at rest have a difference in charge across the membrane? The inside is less positive than the outside. And what happens when we mess that around, and how do we pass a signal across the membrane, down the soma, down the axon, and towards the terminals? That's the question, that's where we're going, and I hope you find it enjoyable. Okay, so I just want to explain a little bit about potential and a little bit about the basics of what we see in terms of the movement of ions across the cell membrane and what it means in terms of potential. So here's a cell membrane with a blue protein channel in it, and at the moment it's closed. We have got 1, 2, 3, 4 positive ions over here, atoms. Now they will in a cell usually be potassium. Let's give it a bit more of a context. And on this side we have 4, again, positive ions, which in a realistic case that would be sodium. Hopefully from the work that we did on atomic structure you can remember what an ion is. And an ion, if you remember, has a positive charge because it's lost electrons. It's got more protons in the nucleus than it has electrons. Anyway, didn't really want to go into that, but hopefully you can remember. Now, if we were to measure the charge across this membrane, so if we were to dump a node in there and measure the difference in charge across the membrane, we can see we've got 4 positives on this side and 4 positives on this side. So actually the charge would be neutral because we've got a balance. We've got the same positivity on either side of the membrane. So let's have a look at what would happen if we open this blue channel and we get blue ions coming across the membrane like so. Hopefully you can see that we get an imbalance in charge now. Ooh, didn't mean to do that. Let's redraw that. So we get an imbalance in charge, and if we were to count the amount of positives up, we'd see 6 positives on this side and 2 positives on this side. Now, if we were to measure the positivity, we would say that this side, let's call it the outside, is more positive than this side, which is the inside. So we use these symbols, the positive and the minus, to identify the, I suppose, the positivity in this case. It's the outside is more positive than the inside. Even though these two molecules here are positive ions, we use the term negative because it's less positive, and that's really the key understanding that you need to start in order to understand this. So positive and negative sides are not necessarily they are negative, it just means they are less positive, which can really flummox some people. Okay. Let's make it a little bit more real in terms of the cell membrane of a neuron. So this is the cell membrane of a neuron, and we're just going to talk about something called resting potential, which means this is the potential across the cell membrane when the neuron is at rest, i.e. when it's not doing anything. It's just sat there waiting to be given a message to pass on. And to cut a long story short, in a neuron, the inside is less positive than the outside. So that must mean we need to lose some of these, positive stuff to the outside, and there's other things that go on like proteins and chlorides, etc. I'm just going to make it simple. Now the inside of the membrane at rest is less positive by minus 70 millivolts, which means it's more positive on the outside by plus 70 millivolts. That's generally the figure that's given. Now different cells have different kind of resting potentials. I think between minus 30 and minus 90 I think can be given, but we're going to use minus 70 as our resting potential for every single neuron in our body. So let's write that, minus 70. And we always talk about inside the cell, so whenever we're talking a figure, we're talking about the inside of the cell, not the outside. We're not really concerned about the outside, we're more concerned about the inside. So let's have a look at really what happens to create this minus 70 millivolts, or this less positivity inside the cell. Here's the inside and outside. So basically what that means is that these sodium channels are always closed, these sodium potassium channels are always open. So what implication does this have? So if these potassium channels are open, what implication does this have for the potassium? Well, you know instinctively that here we've got 2, 4, 6, 7 ions of potassium. So this is a high concentration of potassium. This is a low concentration of potassium. So the natural movement wants, through facilitated diffusion, is they want to pass into the outside of the cell membrane. And that's what happens in order to create this minus 70 millivolts inside. However, because these potassium ions are traveling out, there is another force other than diffusion that wants to put them back in. So they actually want to come back in because this area becomes less positive. They're positive ions, these potassiums. So opposites attract. So because this is a positive ion, they want to come back in because it's negative in here. It's got an electrical force or magnetism force that wants to force it back in. So we actually get a bit of a trade-off. Potassium comes out, but it wants to come back in. And it's that's a major feature in creating this minus 70 millivolts. There's like a tug of war going on that creates this less negativity inside, and that's what creates the resting potential. Now you're not really going to get tested on how resting potential is created, so it's not really fundamental that you're going to have to commit this to memory. You just really need to know that the inside of the cell is less positive than the outside, but it's just nice to know that what's going on at this point is that how it's been created. Another feature is that inside the cell there are negatively charged proteins, which also gives it a less positive charge, if you like. And there's also chloride ions going in, etc. But that's it. That's essentially the main features of the resting potential. And in summary, if you don't get anything other than that, it just relates to the fact that the inside of a cell membrane, particularly a nerve, is less positive than the outside, and the general measurement is minus 70 millivolts. Okay, so this is a zoomed in slide of the membrane on the dendrites and the soma of a neuron. So you should hopefully be able to identify where we are in terms of the nerve. So on the outside of the nerve then, outside of the neuron, I'm drawing these very roughly, but it's surrounded in sodium ions. And this sodium is all around the neuron. So if I zoomed out of this nerve, it would be all around the axon, around the terminals, everywhere. Okay, and on the inside it would be absolutely flooded with the circles, the blue circles which I had on the diagram, which are potassium ions. So nerves are full of potassium and they're soaked around it in a lot of sodium. And we know that there are the potassium channels on this membrane that are allowing them out at rest. And we get this tug of war occurring until no more can move, and we actually get a plus 70 millivolts on the outside and minus 70 millivolts on the inside. And if we were to measure any point on that membrane, at rest, and I'm drawing some lines here, if we were to measure any particular sort of point, if we were to drop in a node here and a node here and measure the difference in voltage, we would see this minus 70 millivolts on the inside and plus 70 millivolts on the outside, and that's resting potential. You need to know that before we move on to how this gets messed up and starts off the nervous impulse. So like I said on the introductory video, the graded potential is really what stimulates or what kicks off a nervous impulse to be fired down the neuron. And a graded potential occurs in the cell body or the soma. Okay, so it occurs across the cell membrane here. That's where we're going to be talking about first. And it depends what type of nerve it is. So it could be a sensory nerve, it could be a relay nerve, or it could be a motor nerve. And it depends what type of nerve will stimulate this graded potential to start off. So if we're talking about a sensory nerve, then we might be talking about some kind of pain, some kind of heat, some kind of touch, anything that this neuron is there to detect. So we've talked about receptors before, baroreceptors, nociceptors, proprioceptors, thermoreceptors, etc. They're all there to detect a certain type of information. So if you touch something right now, you know you've touched it because of the pressure that's been put on that. And this is what kicks off this graded potential. So some kind of stimulus causes a graded potential. Now, if it was a relay nerve, then this might be a relay nerve. If you remember, transmit the information from a sensory nerve to a motor nerve. So this graded potential here on this next nerve could be a signal from this previous nerve onto that nerve there. So it could be this trigger could be like a synapse, it could be a neurotransmitter that's opened up some channels over here. So it's not always stimulus like temperature, pain, heat, etc. It could be stimulus from another nerve that's passing on the signal. Or it could be, for example, in the brain, it could be your brain connecting to a muscle, firing it off to move in a particular way. All right, but you get the picture, hopefully. And so what we're going to be talking about here in this first graded potential is what's going on in the soma. But you do need to be aware that later, when we talk about saltatory conduction, graded potentials do occur wherever there is a myelin sheath or wherever the axon's coated with Schwann cells. Initially, we're going to talk about the soma, and then we'll relate it back to the myelin sheath. So let's draw the soma or the cell body. You have to forgive the crudity of the diagram. Hopefully it will do what I need it to do. So, can you remember what type of ion is surrounding the cell? Pause the video, ask yourself the question, come up with the answer. What is the type of ion that surrounds the neuron? Would be sodium. On our diagrams, we're going to call them little triangles, but they're red on our diagram. Okay. And what are the ions within the soma or the cell? They are potassium. Now, potassium really isn't that involved in the graded potential, but I've just put them there anyway. And what is the charge across the cell membrane? So if I measure the cell voltage or the potential difference in charge across any point in these membranes, what would I see? I'd see that the inside of the cell is less positive than the outside of the cell, anywhere along that cell membrane. Now, let's add another one here. Now graded kicks off. This is the start of where the magic happens. The graded potential has to be stimulated, like I said in the last slide. It needs to be stimulated by temperature, by touch, by another neuron. Something happens that causes sodium channels to open. So I'm putting, these sodium channels are all along the membrane. I've just drawn 2. I could equally draw hundreds of them around the membrane like this. We're surrounded by sodium. But let's say we touch this area here. So some kind of stimulus has stimulated this part of the dendrite. This sodium channel opens, and what will happen? You don't need to be a genius to work that out, because we've done it 100 times. Sodium floods in. Now I'm just going to draw the red dots, because the triangles will get a bit silly. So wherever there is stimulus, so let's say it's touch. Let's say this neuron is sensitive to touch. Whenever we're touching something, these sodium ions will flood in this particular area of that neuron, and then swiftly close because we release our touch. We've just touched something very, very quickly. Okay. What will happen to the charge at this point where the, let's call it point A? What will happen to that point there in terms of charge? Remember, it was -70 here and +70 on the outside. Well, we've just had an influx of positivity from the sodium here. So actually what happens is this charge will become more positive. Now if we're going to become more positive from -70, we don't go to -80 and -90, because that would be becoming more negative. So what actually happens is we go towards 0, and in fact become a positive number. So I'm just going to make this number up, and I'm going to say that this place now becomes +120, and the outside of the cell becomes -120, just in this case. Now it's very important to note at this point in the neuron, it's still -70 on the inside. We're only talking about the area that's just had an influx of sodium. But what we've actually done, if you can see, is we've made now the inside more positive than the outside. We've gone from -70 to +120. So we've gone from being less positive on the inside to now being more positive on the inside, and that's called depolarization. Imagine the north and south poles of our globe being switched around, so the north pole is at the south and the south pole is at the north. We've depoled it, we've depolarized it. We've depolarized the membrane. We've taken it from being less positive to being more positive. Now the sodium that's come in here, the channel has been shut. It has nowhere to go. It can't go outside. So where do these positive ions go? And why do they do that? Well, we've now got a high concentration of sodium. We've got low concentrations of sodium all around the neuron, or the soma, the cell body. So these sodium ions diffuse out, and because they're positive, and this is an area of negativity or less positivity, if you like, here's -70 down here, and here's +120. So they want diffusion force, and the magnetic force from the positives to negatives means that this sodium wants to spread out and spread out. So if we measured the voltage now at let's say this point here, so B, I'm putting that in B, if we measured the voltage, because some of the sodium is actually diffusing down here, where this was -70 on the inside, it could well now reach let's say +10. because this sodium has made it more positive. It's travelled down, but it's not as positive as what it was because all the sodium's here at part A. Some of the sodium's gone west, some of the sodium's gone south, some of the sodium's gone east, and it's only got to plus 10 here. So actually what we're seeing is that this signal over distance has dissipated, has reduced. It's gone from being 120 to plus 10. And if we were to measure the voltage down here, further away from where we had that influx of sodium, it might even be minus 60. You know, it's only gone up 10 millivolts. And by the time it's got to D here, it's back to minus 70. Because the signal weakens as it gets further away. And so what will happen is these sodium ions are pumped out over time. And actually, in this case, nothing would ever happen. So we wouldn't even feel the touch that we've just had. And particular areas of our body that aren't sensitive to touch, possibly we do touch things and we never even feel them. So what is the point? What is the point where I'm trying to say? Well, if we have enough stimulus, so if I'll draw this in a different colour, let's say orange. If we have touch here, if this neuron stimulated here, 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 and here, and here, then not only the sodium channels open at this point on A, but they open here as well, and here, and here. So we get in basically a massive influx of sodium coming into this cell body. Now this makes it super positive. Sorry for the mess, but it's getting super positive here. Where is this sodium going to want to go? And it's the same answer to the question I asked earlier. The sodium wants to go from high to low, and it also wants to go from positive to negative. And the only place that the sodium can go, if I can draw it down here, is the axon. Here we've still got minus 70 and plus 70. Here we have low concentrations of sodium. Here we've got high concentrations of sodium in the cell body. So this sodium is channeled down the axon hillock and into the axon. And if enough sodium gets to this point here, then there's no stopping it. The signal will fire. Because what will be triggered is something called an action potential, or an AP. I've just called it AP, action potential. So I'll just repeat that again. If enough sodium, or if enough stimulation occurs of this soma, and enough sodium has come into the cell body or soma, and enough sodium gets down to this axon hillock, then it will trigger an action potential. And if an action potential is triggered, there is no stopping it. It's all or nothing. The neuron will fire or it won't fire. And that's where we come to the action potential. So before I move on to the action potential, I'd forgot to summarise the two qualities of a graded potential. There are two qualities. One, a graded potential is fast, it's immediate. As soon as those sodium channels open and that sodium's come in, poof, it will go to the areas of low concentration. Poof, it will go to areas of negativity because these sodium ions are positive. They just do it. It's frightening how fast. The second relates to the effect, or the signal, should I say. Let's write that properly. The signal dissipates. Love that word. Or it gets weaker over distance. That relates to the fact that if we've got the cell membrane here, if it's 170 millivolts here, positive, then when we measure it here, it might be 100 millivolts. By the time it gets here, it might be plus 10 millivolts, and by the time it gets here, it might be minus 60 millivolts. So actually, over this signal of the time, the signal gets weaker. It doesn't last forever because the sodium is being pumped out, and the sodium goes other places and diffuses around the cell. It just doesn't go forever. It will run out, and so if you don't signal it enough, if you don't stimulate the nerve enough, nothing will happen. Okay, so they're the two summary facts about a graded potential. So let's talk about the action potential then. And the action potential starts where the graded potential has sort of just got interesting. So here I'm going to draw the axon hillock. And the axon hillock is going to turn into the axon. Okay, so what we've just said is that we've just stimulated the neuron a lot, and that neuron has brought in a lot of sodium, and this sodium has one place to go. It's coming down the axon hillock, down the axon hillock, and we've got a lot of sodium coming down here. So this is where the magic of the action potential starts, because what we've got on the membrane here at the axon hillock, and it's the first time we've seen these, is voltage gates. They're protein channels that are open and closed to certain voltages. Okay, they're programmed almost to open and close when we get to certain charges. Now, these are my sort of opening and closing times in the bottom right. They're not always, accurate. The body has different values, but these are the values that I'm giving, and it's relatively lifelike. Okay, so what we can see there is that the sodium gates in this top rule, is that the sodium gates open at minus 60 millivolts. So here's my nerve at minus 70 millivolts at rest. This is at rest. And unbeknownst to him, at this point, sodium floods down the axon hillock and causes the inside of the nerve to become more positive. So what happens if this nerve, this part here, this protein channel, if the inside of the membrane gets to minus 60 millivolts, because we're adding this positive to the minus 70, then this channel is going to open. The same with this channel down here. Once it gets to minus 60 millivolts, it opens. What is that going to do? Well, we are bathed in sodium ions. So this sodium wants to flood in. Now the channel's open, this sodium is going to flood in. Why? Because it's a positive ion and it's a high concentration. So we've got this diffusion force and this magnetic force wanting to force this sodium into the cell. Okay, and we have the same coming on down here. Now what that will do is it will continue to increase the positivity within inside the cell. So where it started to open at plus 60, this channel is going to get as high as being positive. So it might go to plus 30 millivolts. So the inside will be more positive than the outside, and we've caused depolarization. We've switched the poles. The inside has become more positive than the outside. What implication does that have? Well, all along this membrane, of sodium channels. Now, where does this sodium want to go? It's positive. It's a high concentration. Here, all along this membrane, is minus 70. It's less positive. So we've got that magnetism force again, and we've got that diffusion force that wants to send this sodium down the axon this way. And if enough sodium gets to the next sodium gates and gets to minus 60 millivolts, then the sodium will open, sodium gates will open, sodium will flood in, will cause depolarization, so it'll get to plus 30, minus 30, and enough sodium will come in and cause the next gate to open, and the next gate to open, and the next gate to open, and sodium will flood in and flood in. And so this is where you see that if we get to this magic figure here, this minus 60 millivolts, it's called the threshold. If we get to that figure in this first gate, then there is nothing stopping this neuron from firing the signal all the way down the axon, because sodium gates open at minus 60 millivolts, which is causing depolarization. Hopefully you can see that the sodium gates close at plus 30. So when that's the maximum depolarization that it can get to. So the sodium comes in, gets to plus 30, and the sodium gate shuts, so no more sodium can come in. So actually these gates start shutting one after another as they get to plus 30 millivolts. What you can see on this section here in blue is that we also have potassium gates intermingled with the sodium gates, and they are sensitive to voltage 2. Now look at the rules. Sodium gates open at plus 30. So as this depolarization's occurred, as the inside has become more positive, the potassium gates open this time. Where is the potassium stored? It's inside the cell. So potassium, once depolarization's occurred, the potassium gate opens and lets positive ions out. Now just think about that. At this point in time, at this point here, it was plus 30. But because now we're losing positive potassium ions, it now makes it less positive than the outside. So it will return it back to its minus 70 plus 70 charge. Because we're losing this positive potassium, it will then bring it back down to actually more so than its resting potential. Now because we're going back from being more positive on the inside, and now we're becoming less positive on the inside, this causes repolarization. We're restoring the polarity across the membrane. We're restoring the fact that the inside becomes less positive than the outside. And this continues along the neuron in an action potential. Sodium comes in, potassium comes out. Sodium comes in, potassium comes out. Sodium comes in, potassium comes out. And it will go forever, because these magic figures always are always achieved. One further note is that the potassium gates close at minus 80. So actually it becomes minus 80 before the potassium gates close, and more potassium is left out. So I just want to sort of explain that using this graph here that you can see on the right hand side. This is a graph that can be that can identify, just delete some of this so you can see it a little bit more clearly. This graph identifies the polarity, the charge or the potential at one particular point. So I'm just going to draw one set of sodium and potassium gates just so it might become a little bit clearer, because it did get a bit messy. So what we're saying in this graph here is that minus 70, this number 1 here, or so actually this minus 70 is the resting potential. So this is the neuron at rest. So if we were to put in a measure at this point across the membrane here, M for measure, I'm just going to say we're measuring the potential, then it would flatline at minus 70, and we'd see this sort of minus 70 carry on. But because we're seeing this upstroke in the graph here at number 1, what's causing that upstroke? Well, the sodium from previous part of the neuron is causing this charge to go from minus, it was minus 70, but now it's gone to minus 60 because we've added positive to it, and that's the charge that you can see on this graph, this first part of the graph. Now the dotted line that you can see at minus 60 is this threshold, because once we've got to minus 60, the sodium gates open, and if the sodium gates open, sodium floods in, and that's what you see in this upstroke of a really big upstroke of the graph. That's sodium coming into the cell. That's how I like to draw it on the graph, because it reminds me that sodium is coming in, and that is causing depolarization, because the inside of the membrane is becoming more positive than the inside. Now at 3, we can see in the chart below, delete these, we can see that on the graph the charge has got to plus 30 within the cell, and at plus 30 the sodium gates close and the potassium gates open. So because the potassium gates open, potassium leaves the cell, therefore repolarizing the membrane, i.e. making it less positive on the inside, because the potassium is escaping the cell, and that brings it down to actually lower than the resting potential. It brings it down to minus 80 within the cell, and that's called overshoot. And once that's occurred, so as the messages, as the signal's been put down, the potassium needs to be put back into the cell, the sodium needs to be pumped out of the cell. So through active transport, the resting potential is then reset back to minus 70 millivolts. So the sodium has to be pumped out and the potassium has to be pumped back in, so that another message might fire off and the nerve becomes useful again. So that is the action potential. It's a little bit more complicated than degraded, I would say, but the 2 key facts are written in this top right hand corner. That actually should say action potential, not graded potential. Oops. In fact, that is graded potential, but we're talking about action potentials here, which is stupid. So graded potentials are the opposite. They're relatively slow because they have to go in, out, in, out, in, out, in, out of these protein channels. There's a lot of action going on, but their signal will go on forever, because as soon as you meet these thresholds, or as soon as you meet the threshold, then the gates will open and close no matter what. So that is an action potential. Hopefully I've done it justice. So I've put this slide in hopefully as a bit of a recap on the action potential. So hopefully you can look at the graph and and identify what's going on. It's often a key feature of an exam that you might see this, and you can see I've taken this from an exam, and it might be identify what's going on at the points 1, 2, 3 and 4 along a neuron. So I just wanted to take you through it so that you become familiar with the graph. You can obviously see that there's time along the bottom in milliseconds and the potential along the side. You can see that -70 is where it starts, where the potential difference starts. And you'd be right in assuming that that is resting potential. In orange, then, this upstroke here, the short upstroke, is due to the sodium coming in from previous spaces on the neuron, and it gets to this dotted line here, which is the threshold. What happens at threshold? What happens on this graph at -70? Well, hopefully you can say that the sodium gates open and sodium floods in. Now, because sodium floods into the cell, it causes the charge within the cell to go from -60 to +30. So it actually goes more positive on the inside of the cell, and that, at number 2, is depolarization. Sorry, that looks like a B. Depolarization. Because we've made the inside of the cell membrane more positive than the outside. We've changed the polarity around. Now, I like to imagine this graph, this inside of the graph, as being the inside of the cell. That's why I draw sodium going in causes the charge to go up. And at number 3, the sodium gates close and the potassium gates open. Now, because the potassium is inside the cell, here, inside my cell, it goes out. Because positive goes out, it causes repolarization of the membrane, i.e. it goes back more negative all the way down to -80. That's a repolarization because the potassium ions are going out. Yeah, that's repolarization. We can cause this little thing here. So if that's resting potential, it actually becomes more negative. That's called overshoot. And it just makes sure the neuron isn't oversensitized. It doesn't fire off again. And what has to happen, sodium potassium pump returns the voltage back to resting potential. So it pumps sodium back out and it pumps potassium back in, ready for the nerve to fire off again if a signal is stimulated. So if you remember from the introductory slide, saltatory conduction is the alternation between graded, action, graded, and so on. It's the alternation between the different signals. So what we've got is, let me draw a rough diagram of the soma and the axon hillock. So here it's a graded potential, and at the axon hillock it then turns into an action potential because we've just had those voltage gates and sodium comes in, potassium comes out, and sodium comes in, and that'll happen all the way down the axon. And I'm extending this just to show you what happens. But this neuron is surrounded in most part by the myelin sheath. It's surrounded by it. Okay. Now this axon goes all the way through the myelin sheath, but because the myelin sheath seals the axon, it seals it. There are no sodium and potassium channels for the sodium to get in and for the potassium to go out. So I'd like to zoom in on this part here and this part here. So here we've got the axon, here we've got the myelin sheath. I'm only doing it in 2D. Okay, so what we've got is this sodium coming in, and we've got a lot of sodium that's just flooded in because the action potential's come down, sodium's come in, potassium's gone out, sodium's in, potassium out, sodium in, and so we're getting a lot of sodium because the voltage gate's open, but there's no potassium gates, and there's no way that this sodium can exit. So here we've got, let's say, a plus, what did we say, plus 30, was it plus 30, I think? Plus 30 millivolts. So we've got this depolarization. The inside has become more positive. Now it can't go out of the channels. So where's it going to go? And it's exactly the same principle as what we said. Here's minus 70. This part of the nerve has not been stimulated before. But there's no exit. There's no sort of in and out of sodium and potassium. So it turns into a graded potential. It goes from high to low, it goes from positive to negative. But the beauty of it is, remember, when I did that summary, a graded potential is fast. It's super fast. So the fact that this myelin sheath surrounds it, turning it into a graded potential, it's actually turning it from slow to fast. That's what we want. However, the signal's going to get weaker and weaker and weaker and weaker and weaker and weaker because it's just diffusing all over the place. But here's where the magic occurs. If we have enough sodium around at this point to get the next sodium gate to that threshold figure of minus 60 millivolts, sodium's going to come back in and potassium is going to come out and sodium is going to come back in. So the length of the myelin sheath is just long enough for the sodium to come out of the other end and make sure it triggers the next sodium voltage gate. And if it does, and it always does, it'll boost the signal and get more sodium and potassium coming in before it meets the next myelin sheath. And it does exactly the same. So here we can say it's going from an action potential to a graded potential to an action potential to a graded potential. And there's a definite reason for that. If we zoom out, we can see that one of the reasons why nerves are so useful is because they're so fast. They're so fast, which is why the nerve is covered in this myelin sheath. But it can't be covered in just one myelin sheath because the signal will just be lost down the axon. We have to have these nodes of Ranvier in order to boost the signal. It goes from action potential to graded potential, and the sodium zooms down the myelin sheath, and it just gets enough to make the charge minus 60 for more sodium and more potassium to come in and out, which then zooms down fast down the myelin sheath, which again makes the next sodium gate, voltage gates at minus 60 to open, to cause that threshold to get that in and out of sodium and potassium. So whilst we want the nerve to be as myelinated as possible, we have to have these nodes of Ranvier to boost the signal, to get more sodium and potassium in. If an axon was purely myelinated from start to finish, there's a good chance that this signal will not be passed on by the time it gets to the end of the myelin sheath because the signal's dissipated. And that is the function of the nodes of Ranvier, is to boost the signal, is to allow an action potential, a very short action potential, to kick in order to then zoom more sodium down the other end of the tunnel, if you like. So saltatory conduction is the switch between action and graded potentials in order to make sure, it's a trade-off, it's to make sure the nerve is as fast as possible, the signal is as fast as possible, but in order to make sure that it definitely gets there. Okay, and it takes advantage of the speed of a graded potential, but I suppose the infiniteness or the distance of an action potential. Likewise, you wouldn't want an axon not myelinated. So the sodium comes in, potassium goes out, sodium in, potassium out. And so can you see how long that would take, sodium in, potassium out, sodium in, potassium, in comparison to a myelinated section where it goes zoom, straight down, in, out, in, out, in, out, zoom, in, out, in, out, in, out. And that's how a nerve is propagated, or a signal is propagated along an axon. How magical is that? How insane is that? But how beautiful is it, just to make sure that that signal gets from one side to the other. And that's how your brain is thinking. That's how you're sending signals to your muscles to move. That's how your brain is signaling messages to your heart to speed up, to slow down. You know, it's keeping us alive. And all it comes down to is the in and out of this sodium and potassium. I just wanted to finish off with a bit of sort of implication in terms of pharmacology. And you may well have wondered, you sat at the dentist and you've probably unfortunately had an injection into the gum, but fantastically you don't feel the dentist drilling around and scratching and scraping, and you don't feel any pain. And likewise, you may well have gone or had an unfortunate event where you've had to have an operation under general anaesthetic. How does that stop the pain but yet make you unconscious so you don't feel anything? Well, it comes down to the structure of the neuron, and you've got two, essentially, I suppose three areas in which you can affect the activity of a neuron, forgive the crudity of the diagram. You've got three ways of killing pain. We're just going to talk about pain, but we can talk about other stuff as well in terms of nerves and how we can affect them. In one hand, you can stop the stimulus. So you can stop the thing triggering the nerve off. So drugs such as ibuprofen stop inflammation. They are an enzyme inhibitor, I think, off the top of my head. So they stop a molecule being made which fires off the pain signal. So if we can stop the dendrites being triggered to start that graded potential, then we're never going to get this threshold at the axon hillock. So that's how possibly a paracetamol or ibuprofen works. The difference between ibuprofen works at the local area, so it stops the inflammation and swelling and therefore stops the pain signal, whereas the paracetamol works in the central nervous system, the spine and the brain, and acts on neurons in a different way. Similar but different. Anesthetics work by causing these sodium and potassium gates along the axon to not work. So if you can imagine, if you've got sodium gates there that need to get to the threshold and you're blocking them and you're not letting this sodium come in, then you're never going to get to the threshold and you're never ever going to get this signal. It completely and utterly blocks the nerves within that region. Now you've got different anesthetics and you've got local and general anesthetics, but they generally work the same way. They stop the sodium and potassium gates opening and closing. If you do that, you're never ever going to get a message sent down that nerve. And all pain is an interpretation of a signal from a part of your body. So if you feel pain, that signal goes to your brain, it goes to the thalamus, I think, and then it gets channeled into other areas of the brain to be made sense of. So sometimes we feel pain, that signal goes up to our thalamus, the thalamus channels it into some area of our brain for memory, let's say, and we remember that pain is not life-threatening, okay, we've stood on a pin, we're going to get better, and therefore we don't have really an emotional reaction, we don't have a fight or flight response. Whereas you break your leg, for example, it's a pretty serious deal. So that signal then will be channeled into a part of the brain, which then might have a different reaction, and that's how these neurons, you get a flavour of how these neurons might work. But pain is just a signal. So if you stop the signal from getting to the brain, you don't feel pain. There are certain people with genetic conditions that don't, their neurons, their nociceptors, so nociception is the detection of pain, they don't work. So people bite their tongues off, they break bones just because they can, and they don't even feel it, they don't even recognize it, so they can cause some serious damage to themselves. Now the third possible way is you can stop this message being transmitted by the terminals. So that would be inhibitors at the synapse, for example. So if you've got signals coming down, if it stops at the synapse, it's never going to pass on to the next nerve, and therefore you might not feel pain. And there are blockers of neurons that do that. So you've got different ways in terms of pharmacology that acts on nerves, either at the head, the axon, or the terminals. And that might unlock a bit of reading for you. I mean, you might be able to look at exactly how certain drugs work if you're interested. You might be thinking about members of your family, or you going to the dentist and thinking, well, exactly how does my gum feel numb? And you can easily go and find that information now, having known how a nerve works. So yeah, hopefully it's been useful. Hopefully you've enjoyed it. I know it's taken a long time, but I probably take it 3 lessons at least, 3 lessons going through this. So at least you can replay it to your own heart's content, and we'll develop this further in our next session.